Hello everyone, welcome to my channel. I'm Eason from the Department of Chemistry at UCL, and today I'm going to take you on a fascinating scientific journey exploring how DNA origami may hold the key to a new solution for diabetes. Diabetes is a prevalent and serious metabolic disease characterized by persistent high blood sugar levels. In a healthy state, our bodies have a sophisticated blood glucose regulation system. First, beta cells in the pancreas secrete insulin. Then, insulin acts on insulin receptors. Finally, cells absorb glucose from the bloodstream, lowering blood sugar levels. However, in pathological conditions, this system have changed. For type 1 diabetes, beta cells are damaged and cannot produce sufficient insulin. And for type 2 diabetes, although insulin is produced normally, cells cannot accept it. So in both of these two cases, insulin fails to function properly, leading to chronic high blood sugar levels. If left uncontrolled, diabetes can lead to a series of severe complications. And these complications seriously threaten patients' quality of life. So what is the treatment of diabetes? As we previously discussed, the key point of diabetes treatment is to control blood sugar levels and increase insulin secretion. There are several medications currently used to treat diabetes. And I want to introduce this one to you. It's called exenatide. It's a drug that's widely used to treat type 2 diabetes, and it's a GLP-1 receptor agonist. Before we introduce our new targets for diabetes, let's first look at how this drug works. So this picture shows there is a GLP-1 receptor exists on the membranes of beta cells in pancreas. And GLP-1 is an endogenous ligand, and it acts as a key to interact with GLP-1 receptor and exert its biological function, that's to increase insulin secretion. And scientists have discovered a drug that's named as GLP-1 receptor agonist, which can mimic the function of GLP-1. So it can also interact with GLP-1 receptor and exert the same biological function. So in this case, we use this receptor as a drug target, and we target protein. But today, I want to introduce something new, and this time we don't target protein, we target the gene promoter region of insulin gene. Before we move on to our new targets, let's first introduce the transcription process, which is quite essential for gene expression. In this picture, we can see that upstream of the coding sequence, which transcribe DNA to RNA, there is a DNA sequence called promoter, which is quite essential for transcription. And in this picture, we can see the transcription process more clearly, that RNA polymerase II binds to promoter regions, and also it binds with transcription factors, and then it initiates the gene transcription. But what's important here is that upstream of the gene transcript, we notice a gene sequence that's called enhancer in this orange line. And this is a very special regulatory sequence that can enhance gene transcription. And more excitingly, scientists have discovered a special enhancer sequence in the promoter region upstream of the insulin gene that significantly enhances insulin gene expression. And this sequence, give it a name, is called insulin-linked polymorphic region, ILPR. And it holds promise as an innovative target for diabetes treatment. ILPR is also known for its unusual DNA structure. Now, let's discuss it more clearly. Before we move on to the mysterious structure of ILPR, let's first introduce this typical DNA structure. That's the double helix structure that we are all familiar with, which was discovered by Watson and Crick in 1953. That two DNA strands intertwined like ropes and connected through base pairings, A with T and C with G. But in this case, the ILPR sequence reveals the astonishing diversity of DNA structures. Scientists have found that ILPR contains numerous consecutive C and G bases, which prompt DNA to form atypical four-stranded DNA structures. That's the quadruple helix. And one example is called I-motif. And this is formed by sequence which rich in cytosine base pairs. And these C-rich DNA fragments combine through C base pairs with heavy proteinated C base pairs. That's quite different from the previous base pairing as we previously discussed. And the second unique DNA structure is called G quadruplexes. And in this case, four G bases form a G tetrad through hydrogen bonding. And they stack like building blocks one by one. And it is stabilized by potassium ions. And these four-stranded structures aren't just scientific curiosities. They do play key roles in regulating gene expression, and it is also as a inspiring new targets for diabetes treatment. Scientists have discovered that within the human population, the ILPR sequence is very different and shows a lot of change. And you can see that in this table, from 1G to 6G. These repeat sequences differ from each other. And you might think that these G-rich sequences might also form G quadruplexes structures, but actually it's not true. That only 3 of them can form G quadruplexes structures. And what's more interesting is that the repeat sequences which can form G-quadruplex structures, also shows that it can activate the promoter activity. That's what we call acts as an enhancer. And also, the most structurally stable, the most stable structure is this one, 1G. And this repeat sequence shows the most structural stable variant and is also closely associated with the highest promoter activity. So this discovery offers a fresh approach to dye-based treatment. We can design small molecules to target these quadrihelix structures and to stabilize these structures, thereby activating promoter activity and promoting insulin gene transcription. This is like turning on the insulin production switch at its source. That's a quite completely novel therapeutic strategy for diabetes treatment. However, there are still some challenges when target ALPR as targets. First is the dynamic nature of nucleic acid structures. The I-motif is quite sensitive to pH. And a slight change of pH might influence the interaction between the ligand and I-motif. And the second challenge is that it's quite difficult to develop specific ligand. Scientists need to take everything into consideration, and maybe at first we have thousands of chemical compounds, but after this filter, maybe only a little compounds can act as drug candidates. Nevertheless, the research direction is still, full of promise to target to target I-motif and G quadruplexes as drug target. And we have reason to believe that these remarkable DNA quadruple helix structures will bring breakthrough process in diabetes treatment. And thank you for your listen. Today we've explored a new solution for diabetes hidden in the fascinating world of DNA origami. I hope you like this video and thank you a lot. And I wish you have a wonderful day. Thank you.